am Flavia, and I'm a postdoc in the Delmore Lab and, at Texas A&M, and I'm going to talk about genomic shock in the Swiss and thrush hybrids. So, oh. So, as we expect, maybe just click. There it is. So genomic shock was first reported by Barbara McClintock in the 80s in corn, yes, in corn, and I'm going to talk about birds. And this basically was reported as when you have, as you can see in this small illustration, you have two species with different epigenetic profiles, basically different accessibility in the genome, different T composition, and then during hybridization you have these combined things, but at the same time something happens during hybridization that you will have more accessibility, and then you will have all these combining TEs being expressed, being mobilized, and reactivated. Some of them were like efficiently suppressed in the parental forms, so this is what we see as a genomic shock. These hypotheses have been tested successfully sometimes, not always, and it seems that there are like some TE families that are more involved in this phenomena. So let's see what happened in the Swiss and thrush. So we're going to use this wonderful songbird, a migratory songbird, that thanks to wonderful work and experimental design in the Delmore Lab, we know a lot of things. And one of these things is we have hybrid zone, we have these subspecies that have different migratory routes. You can see here, one goes through the coast, the other one to the inland. You can see on those roads a hybrid zone. We know that they are genetically distinct and they're reproductively isolated. We also know, just recently, something very cool, that there's like a transgressive gene expression in two brain regions related to migration. Here in this, in your right, you can see this transgressive expression as the yellow in different seasons. I will talk about that experiment next. But you can see these two brain regions and others. So these two brain regions, hippocampus is related to like memory information, location, and the cluster N is more related to something that is the geomagnetic compass of nocturnal. So this is very important for a nocturnal migrant, just as the Swiss and thrush. So this work that I'm going to show you today, it's a continuation of this previous published work. And basically is we have these two brain regions that I just explained to you, and this experimental design that basically the Delmore Lab took free-living birds and put it on the lab and stimulated, photo-stimulated to mimic the seasons in the lab. Then we took these like brain punches, RNA-seq data to get the transcriptomes, and ATAC-seq data to get the epigenomes. So we have these states of let's say seasons or non-migrant to migrant state. This was done like gradually, of course. And then we have this divide. We have the coastal, the inland, that I will be talking that way. We have the hybrids, of course. So to test this hypothesis, I went to 3 steps. And the first one, we asked whether the hybrids were expressing high levels of TEs. And the second one, of course, more accessible regions in the genome, or more overall accessibility, or open chromatins. And the third one, we tried to connect these 2 dimensions. And we were, so in the plot on the right, we have this prediction where we expect, you can see on the y-axis, like more different accessibility between the comparisons of the hybrids with the parental forms, and also more TE expressed overall. And when you compare the parental forms, you don't expect to see that correlation as strong. But of course, you will see some correlation, because we know that when the genome is more accessible, in general, you will have more genes to be expressed, more TEs to be expressed, you know. So let's see. let's go to the first step result. So here I first calculate the relative abundance according to the chromosome, right? Like the counts of TEs expressed relatively abundant on each chromosome. So this plot is showing the differences between the hybrids and the parental forms. So imagine that every of each red square is that box plot on the right, where you have the proportion of TEs per chromosome, one TE, one super family, and then you have like the taxon on the x-axis. And the blue squares will be the opposite. So you don't see the opposite, like let's say hybrids lower expressing T's as much, but you actually see. And to give some context, when you see this heat plot where you have the transposable elements on the y-axis, I just put it as major classes to like for practical purposes, and the chromosomes on the x-axis. And for more context, right, to have more perspective, I include also the presence of TEs that were on the transcriptome that are that were not giving like any significant difference between the hybrids and the parental forms. Those are the white squares, and the gray were like squares that were not even present on those chromosomes. So if we see this, so this I'm showing just like the spring in the cluster N hyperpallium. And if we see the overall experimental design where we have these two brain regions, so on the top we have a cluster N hyperpallium brain region, and on the right top we have the hippocampus dorsal, and then on the top we have springs, and then on the bottom we have winter. So overall you see that there are like several TEs that have been like highly expressed or more abundant in certain chromosomes, not on all of them. But also we know, we can see that there is something else, right? Maybe you can detect that there are less squares on cluster N for winter, and the opposite for the hippocampus. So let's go to the step 2. Here we are looking into the accessibility of the genome. And I'm showing you here the hippocampus for the hybrid and coastal comparison just during spring. And on the right, on the left plot, you can see the false discovery rate on the y-axis, the fold change of these like differences between the hybrid and the coastal on the x-axis. And you can see there's a lot of red dots on the part of accessible, which means hybrids are having more accessible genome or points on the genome, peaks, regions, fractions, the way you want to see it. And if I take these plots, just to make it more easy to see, I have this bar plot where you can see that hybrids had more accessible peaks or regions in the order of hundreds compared to non-accessible, but there are also differences in non-accessibility. Now, if we see it overall, these two brain areas and between the parental form comparison coastal and the hybrids, and you can see that both brain regions have the same pattern, right? So we have more accessibility in hybrids. You can see it like they're on the top right. And then if we compare just the parental forms, there's just like few different accessible regions. There are some more in the cluster N, but very few on the hippocampus. So I don't want to show you like and bore you like about tons of volcano plots. So I try to summarize these red dots of these several analysis comparisons in just bar plots, and just focusing on the accessibility because this is important for the hypothesis testing. So you can see in these bar plots the number of normalized or number of accessibility peaks on the hybrids compared to the coastal you see in color, but also the parental forms in gray. So something interesting to see is that on spring, if you focus on the axis of the y-axis, you will see that there are like much more higher differences going on the order of thousands different accessible peaks. Obviously, comparing with the hybrids and the coastal and the inland, you can see there's like much more in the inland comparison between in both brain regions. But on the winter, something is happening. like on the non-migratory state. So here you see the hippocampus is like showing the same pattern, but something interesting is going on for cluster N, and we are still working on the data. Any comments are obviously welcome, but you can imagine, like, we are talking about brain areas, they are not far away, and they must interact. So there must be some conflict. We are trying to elucidate what kind of conflict could be in here, like, in spring you are basically more similar as an hybrid to a coastal parent, and in winter you are like more similar to the island. So this is a very interesting thing just to look at. And finally, for the step 3, where we try to put together these two dimensions of the genomes, I show you here, like on the y-axis, you have the number of accessible regions that it will be like in the hybrid, and on the x-axis, you will have the number of TE differentially expressed on those chromosomes. So each point is a chromosome. And I'm showing you here just the winter, which is showing the strongest association. And when you compare the parental forms, that will be the gray there on the bottom, there's like very few chromosomes, such as the 42, like, which is the sex chromosome, showing like a slightly more, I don't know, like being there, you know, just the fact being there, we don't have too much to compare, honestly. And for the inland, something else also is happening. Association is there, just in very few chromosomes, but is like showing. So now, let's see the overall picture and of the whole experimental design. So we see we can hold the idea of the pattern where the hybrids are showing indeed more accessibility, more TEs being expressed, and the correlation is higher. While there's correlation as well, good correlation for the inland and coastal, something that is expected, we see like few chromosomes being different. So hopefully with this you can see that genomic shock is likely there in birds, in this specific system, and is besides just like the genomic shock hypothesis, when you see the plot on the bottom right, you see the hippocampus dorsal in winter. Well, I know you are thinking like, but Flavia, what's happening there? Like, I know, I thought the same. Something is happening there. We have less differentiation in accessibility in TE express. We are still looking at it. There's probably a more conserved brain region. We're still like working on that. Happy to hear some comments. But hopefully I have convinced you that genomic shock is there, and that hybrids are like in effect showing higher TE transcription in several TE classes and superfamilies. Also more open, more accessible genome, a stronger association. And also I think I want to point here that just by looking this complexity is this association, but also some differentiation and complexity, how the mobilome, you know, of the genome, the transcript, the transposable elements interact with the genomic architecture, different seasons, you know, and in different brain regions as well. So hopefully I have shown you good evidence of this. Can I go through some questions, maybe, or not? Yeah? I have a minute, so please. Questions? Yes?
Are there any comparative effects of this genomic shock? Does it have any impact on the brain?
Good question. We still don't know. It's a good question. I cannot tell you, honestly, if there's like a fitness. We work in the lab on this a lot. There's a lot of ecological work, and to see if there's like some movements that hybrids are like changing, but I don't have like strong evidence for now to tell you.
Yeah, welcome to this talk. My name is Jan Lifjeld. I come from Norway, University of Oslo, and I'm interested in sperm and speciation and birds. And of course, when we try to understand speciation, we need to understand those traits that are actually driving speciation and causing reproductive isolation. And what is more logical than look at those traits that are actually very active in or central in causing assortative mating or assortative fertilization. And of course, sperm is a central player in fertilization. And what is the definition of speciation? Phenotypes? Well, there is a definition here that there are traits that whose divergence contributes to reduction of gene flow or causes reproductive isolation. So divergence is very interesting to start with here, and just go rapidly through a sort of a sequence of speciation where you have a starting point with a population, with a trait, with a mean and a variance, and there is a splitting event, and the traits diverge at a certain speed. You have two means, and as time goes by from T1 to T2, they diverge further, and you have overlap at some point. And the interesting thing that is often overlooked is the importance of variance, because if you see these two populations, they diverge with a mean at the same rate over time, and the overlap is reached faster if you have a reduced variance. So variance is an important part of this. Of course, time is important. The speed at which the means move apart is important, but also the variance. So if sperm and sperm length, which I'm talking about, can function as a speciation phenotype and cause reproductive isolation, there are two assumptions here. First, you must have fast divergence in the trait. So within a species that actually you get separation in the trait in a reasonably short time, and that there is selection on the trait, so that when two populations actually meet again, that there is selection to keep them apart, that you have assortative mating or assortative fertilization in terms of sperm then. And then, if you have stronger selection caused by, for instance, sperm competition or female promiscuity driving sperm competition, then will then divergence happen faster? That's what I'm going to talk about. So what's the evidence for these assumptions? Well, we have a lot of, or not a lot, but we have some cases where we have divergence in within what we can call a speciation continuum, where you have species that are on different stages of their speciation continuum and have different levels of overlap. Here are two examples from birds. The salt marsh sparrows, Nelson sparrows to the left, two subspecies of barn swallows to the right. And you see, you can measure overlap here, either by the amount of variation that is covered by the two curves, or you can use effect size metrics like Hedges G or Cohen's D to say something about how different the two curves are. Hedges G is a simple measure. It's just the difference between the means scaled over the standard deviation of the trait. So that's what I'm using in my analysis. And then about the second assumption about selection. Is there selection on sperm length in birds? Yes, there is. And several studies, comparative studies, have shown that there is a relationship between the variance in sperm length in a population and the level of sperm competition in the same population. So these are points are from 24 species where we have measured the level of extra pair paternity as an index of sperm competition or female promiscuity on the y-axis, and the CV of the sperm length in the same population, and there is a very, very strong negative relationship here, which means that there is some selection going on when we interpret it as intensity or the strength of stabilizing selection caused by female promiscuity reduces the variance in the trait. And remember one thing, that the trait, sperm length, is, when it's under selection, it's under selection to perform in the female environment. So the relationship between the female oviduct and the sperm is, of course, very intimate. And there's this classical paper from the 90s by Jim Brisky and Bob Montgomery, showing that sperm length in a species is highly correlated with the length of the sperm storage tubules in females. So there is a very strong co-evolution between the sexes here, and that the sperm is adapted or selected in the environment of the female. So if there is any change in the sperm storage tubule length of the female, strong stabilizing selection by female promiscuity will actually make the sperm adapt very rapidly. So there is selection on sperm length in population, which is actually related to the intensity of sperm competition or female promiscuity. So does sperm length then diverge faster when females are more promiscuous? So to answer that question, we collected the data set with 20 pairs, population pairs, with divergence times within 2.5 million years. So these are populations of the same taxonomic subspecies or subspecies of same species, or species pairs that have newly been elevated. You know, taxonomy can be not stable, but or it could be generic species that still have a hybrid zone. So there is different levels of this where they are on the speciation continuum. So this is the data set actually illustrated here, and you can't read perhaps the names there, but we have 20 pairs of closely related populations at different stages of divergence. And to the right, we have the sperm data. So we need to have sperm length data for the two populations and their variance. We need to have the divergence time, which is calculated in these cases by nuclear or mitochondrial DNA kind of estimation. And we need estimates of promiscuity, which is in the brackets for the pairs here. And see here all the variation in sperm length. So sperm length is a trait that is actually very variable across the songbirds. So when you have this huge diversity, it's for me, it's quite logical to think, oh, could it be sort of associated here with the speciation and this very, very diverse trait, which is related to, strongly related to the fertilization process. Yeah. So we have then three variables: divergence time, we have the divergence in the trait, and we have promiscuity. So here on the y-axis, we calculated the divergence. It's a log scale though, but using Hedges G. So, and on the x-axis here to the left is the number of generations or the divergence time. And there is no relationship because this divergence in sperm traits is much more strongly driven by promiscuity than by the time actually. So if we actually do a linear regression or a multiple regression here and put both variables, both the time divergence and the promiscuity is actually strongly correlated with divergence, so it explains the divergence very well. But if you take divergence divided by the divergence time, we get a sort of an estimate of the speed of divergence, and it's driven by promiscuity. And remember that the Hedges G here is actually a ratio. So it's the difference between the means scaled over the standard deviation. So then the question is this effect of promiscuity does it affect, or is it explained by the difference in the means or by the variance? Oh, sorry. I should have. How do I go back? Where's the arrow here? There. Yeah. So, so then we have promiscuity on the x-axis, and then the difference between the means, and then the standard deviation. And you see here there is a positive correlation with the explaining the mean, but not significant. But it's mostly driven by the standard deviation. So the divergence is or the effect of promiscuity is because these populations have low variance in the trait. So overlap happens much earlier when you have low variance in the trait caused by by promiscuity. So it follows this pattern of trait variance. So the high promiscuity gets non-overlap faster. So speciation can actually happen more rapidly when you have high promiscuity. So that's my conclusion. Sperm length diverges faster in more promiscuous species. And that's because of reduced variance, and not because the means move faster, diverge faster. So what I've shown here is actually the sort of the assumptions of the hypothesis, but the unanswered question so far, what does that mean in the contact zone? When you have two optima that meets, it means two female environments that sperm has to sort of meet with two females. Is it so that you have reproductive isolation in the hybrid zone based on sperm length? We need to show that. So that's my challenge to you. If you have good hybrid zones with sperm length divergence, look at assortative mating, assortative fertilization. If that's true, then we would also predict on the macroevolutionary level that more promiscuous lineages have higher species richness because speciation goes faster. That is also interesting to test. And if that's true again, then maybe some birds, which is a promiscuous radiation, maybe it's a gametic radiation instead of what we normally think of it as an adaptive radiation. Okay, thank you.
Hi, I forgot to put my name on the slide, but I'm Steph Blain, and I'm a postdoc in Kira Delmore's lab at Texas A&M University. And I'm going to talk about repeatable selection on large ancestry blocks in an avian hybrid zone. So hybridization is, I think, an interesting evolutionary process. One of the reasons being that with hybridization, we get the combination of diverged genetic backgrounds in one genome. So thinking about F1 hybrids, we get sort of complete heterozygosity at sites in the genome that differentiate the two species. But things get more interesting in later generation hybrids when you start to get recombination and kind of different forms of mixing of those two genomes. And what can happen with that is that genomes end up mixing non-randomly. We can get a skew towards one species with the sort of left highlighted section, or a skew towards the other species, or an absence of heterozygosity, or it doesn't matter which species ancestry you have, you just can't have both. And these patterns can arise due to either selection or genetic drift, or various other things. But one approach that we can use to try to infer that it's more likely to be selection is if we observe the same patterns repeatedly. So if you have kind of a few replicate populations, and in this example, we're seeing, kind of introgression of one species background repeatedly, it's more likely to have not been a result of drift. So I'm using hybridization genomics as an approach to look at these processes in the Swainson thrush. Flavia already introduced them, but just to recap, we have two subspecies that are ecologically divergent. They form a migratory divide with one subspecies overwintering in Central America, the other overwintering in South America, and they form a hybrid zone that's quite long. It's very narrow, which provides evidence that there's selection acting on the hybrids in that zone. And it, kind of runs the length of the Pacific coast. So we sampled in 4 replicate hybrid populations across the length of that hybrid zone, which you can see on the map on the left. The histograms on the right just show distributions of genome-wide ancestry in those 4 populations, and then also in 2 parental populations. So you can see that there's some variation in genome-wide ancestry in all populations, including some sort of later generation back crosses and that sort of thing. And we can see that with a finer scale when we look at ancestry informative markers. So we did low coverage, whole genome sequencing, defined ancestry informative markers, which are sites in the genome that are quite differentiated between the 2 subspecies, called those in hybrids. And here's just a few representative individuals from each population. Each bar is one individual, and the color indicates subspecies, with blue being coastal, yellow being inland, green being heterozygous. And you can see that there's a lot of variation within and among individuals. So this isn't a recent hybrid zone. There's been a lot of admixture here for a long time. Okay, so using that data, I looked at repeatability across the 4 populations of some patterns that we considered neat, because they might be representative of processes like selection. So for selection, I looked at genomic Klein gradients and ancestry transitions, and then introgression, genomic Klein gradient, or genomic Klein asymmetry, and then FD. So starting with the genomic Klein gradients, so here, genome-wide ancestry is on the x-axis. The probability of coastal ancestry is on the y. Each line is one locus of our ancestry informative markers. So our neutral expectation here is basically a 1-to-1 line. What we're interested in, though, are loci where there's a fast transition from a low probability of coastal ancestry to a high probability of coastal ancestry, because that likely means that there's selection against heterozygotes. And we find some evidence for those fast transitions in all 4 populations. And then we looked at whether there's repeatability across the 4 populations in which loci show fast ancestry transitions. So on this plot, genome-wide or genome position is on the x-axis, Klein gradients on the y. Each point is one locus, with the actual number being the mean across the 4 populations. And then color indicates the number of populations in which a locus was an outlier for genomic Klein gradient. So the pattern that really jumps out here is that there's a genomic block on chromosome 1 and another one on chromosome 5, where there's sort of repeated genomic Klein outliers in 2 to 4 populations. So that suggests that something interesting is happening in those regions. We also looked at the frequency of ancestry transitions, which are places in the genome where ancestry state changes, perhaps from coastal to heterozygous or coastal to inland. So there's variation across the genome in how frequent these transitions are. So some regions have a high rate of ancestry transitions, like the highlighted bar on the left. Others have much fewer transitions, and fewer transitions suggest potentially stronger selection against heterozygotes. The idea here is that there's reduced heterozygosity near incompatibilities in the genome, and this offers fewer opportunities for a combination between the 2 species backgrounds. And this is a histogram of the number of ancestry transitions in 100 kb windows across macrochromosomes, with transitions from the 4 populations stacked. So again, I want to draw your attention to chromosomes 1 and 5, where there's really a valley in the number of ancestry transitions happening in a couple of regions there. Whereas at other parts of the genome, there does seem to be a lot of transitions between ancestry states. Okay, so moving on to introgression and going back to Klein's, here we were interested in Klein asymmetry. So before I was talking about the rate of transition, here I'm interested in sort of where that transition happens. If it's shifted to the left, that suggests that there's coastal introgression. Basically, you get a higher probability of coastal ancestry at a locus than you would expect given the genome-wide ancestry. The opposite if it's shifted to the right for inland introgression. And we only found evidence for introgression in 2 of our 4 populations, the 2 towards the center of the hybrid zone latitudinally in British Columbia, in Hope and Pemberton. But we looked at overlap between those 2 populations in Klein asymmetry. So this is the same as the other plot for Klein's, except here I'm showing the median Klein, or mean Klein center across the 4 populations. The null expectation is 0.5. And you can see that there's some overlap, again, between the 2 populations, particularly on chromosome 1. And in the regions that on chromosome 1 and 5 that I highlighted for genomic Klein introgressions, one seems to be shifted on average in an inland direction, whereas the other is shifted on average in a more coastal direction. Okay, and then as a second measure of introgression that didn't directly involve the hybrid populations at all, we used FD. So this is with subspecies populations that are parapatric to the hybrid zone, and involves looking at the relative frequencies of gene trees that don't match the species tree. So for our species tree, we expect that the inland populations from BC should cluster with inland populations from Quebec because they're the same subspecies. And then the coastal populations are an outgroup to those. But other gene trees obviously are observed due to various processes, including incomplete lineage sorting, including where inland BC populations cluster with coastals or inland Quebecs cluster with coastals. But if hybridization is occurring between the inland BC and coastal populations, we expect to see the hybridization tree much more frequently. Unsurprisingly, this is what we see. We have a hybrid zone. It's not exactly a shock that there's some introgression here. But what's maybe more interesting is that there's some variation across the genome in where that introgression occurs. So for example, if you look at chromosome 3, there seems to be sort of a valley in introgression towards the center of the chromosome. And I didn't look at repeatability here because it's just the one comparison. Okay, so we have some idea that there's sort of at least repeatable patterns occurring. And this repeatability can be due to sort of the same patterns of selection happening on all of these populations. That's likely playing a role. But it can also be driven by kind of genomic structural features, including recombination rate. There are several other factors that might be important, such as gene density, chromosome size, and the level of species divergence, but for this talk, I'm just going to focus on recombination. So for recombination specifically, the idea is that in regions of low recombination, there's much higher linkage disequilibrium among loci, and so that could lead to sort of increased linked selection between loci in that region. So if you're looking at a locus, it's going to have potentially a much stronger kind of apparent response to selection because of that. So because of that, our expectations are that for our metrics of selection, that those should show a negative relationship to recombination rate. And then for our metrics of introgression, if introgression and admixture is generally speaking a bad thing in the hybrid zone, then we would expect that to show a positive relationship to recombination rate. So starting with selection, looking at our genomic Klein gradients, it's a super messy plot, but there's some variation between the 4 populations, but overall we find kind of a generally weakly negative slope there. And then ancestry transitions, I've just flipped the y-axis there, because fewer ancestry transitions is more selection. Anyway, unsurprisingly, there's a strong relationship between ancestry transitions and recombination rate, but that one makes sense. And then moving on to introgression, for Klein asymmetry, kind of similarly to the Klein gradients, we find the predicted pattern with some variation among populations. And it's kind of a weak relationship. And then looking at FD, we find a stronger positive relationship to recombination rate, as expected. So to wrap up, we found a couple of large genomic blocks that we're quite interested in that appear to be under repeated selection kind of across the hybrid zone. One's on chromosome 1, seems to be shifted towards inland ancestry. The other's on chromosome 5, seems to be shifted in a coastal direction. Recombination rate is likely playing a role in structuring the repeatability of these patterns, as are other genome structural features. And with that, I'd just like to thank my co-authors and various others who I've talked to about science and thrushes. Yes.
So you had the hybrid zone as anything within the inland piece, as opposed to, for a lot of species, the Rocky Mountains are considered the sort of divide between species as the geographic barrier.
Yeah.
So I would have expected that inland BC would more align with the coastal BC as opposed to the Quebec subspecies.
Yeah, no, it's a little weird in some ways, because, yeah, basically what happens is there are these birds that fly all the way from, like, the coastal mountains right next to Vancouver, or I guess the Cascades, basically, all the way, you know, east via sometimes North Carolina down to Columbia. So I don't know why it evolved, but it did.
Okay, thank you.
Bonjour tout le monde, je m'appelle Jesse. I'm going to be talking about random walks and potentially how they might be applicable to speciation. And to start out, I'd actually, and this is maybe kind of selfish, but I'd love to just take an informal poll. If you guys want to think about, you know, speciation phenotype of your favorite organism, what's the dimensionality of that space? So imagine that, you know, if you work on arthropods, if you work on insects, for example, the traits that are involved in reproductive compatibility, population cohesion, how many traits is that? How many people think it's less than 4? How many people, and maybe we could say like independently evolving traits, right? So maybe there's just one super trait, right? So how many people think it's less than 4? How about less than 10? More than 10? Okay, that's I'm gonna, 1, 2, 3, 4, 5. I'm gonna include that in the paper. So I'll give you some consent forms to sign after the talk. So I think a lot of us, when we're thinking about divergence in allopatry, the null hypothesis that we use either implicitly or explicitly, you know, as a thought experiment, is this kind of random walk. And I think I'm including this because Futoyama had this figure in several editions of his textbook, and it's a really darkly hilarious cartoon if you kind of deconstruct what's happening, because there is that absorbing barrier where the drunkard presumably dies, I guess, at the end. We're not actually going to be talking about allele fixation in this talk, but I think for a lot of evolutionary biologists, this is how we kind of set our understanding, you know, our kind of mental picture of what's happening in a random walk. But mathematicians have really long appreciated that the dimensionality of random walks has really fundamental impacts on the way they behave in space. And so there are these properties. There's this property of random walks that are referred to as recurrent, which means that if they start at a location and you let them run to infinity, they'll return to that location at least once with probability 1. But then random walks in higher dimensions are referred to as transient. And that means that there's a less than 1, there's a positive probability that even if you let them run to infinity, they'll never return to when they started. There's also really cool properties of random walks that change with dimensionality. Brownian motion, people have studied the probability of intersection of two different Brownian motion processes, and if their dimension is 3 or less, they'll intersect eventually. But if their dimension is 4 or greater, they actually might never intersect. And one cool way of thinking about transience versus recurrence is this just unforgettable quote by Shizuo Kakutani: "A drunk man might find his way home, but a drunk bird could get lost forever." And so I think this is really important to consider when we're thinking about what our null hypothesis is for divergence in allopatry, because the phenotypic space that controls reproductive compatibility, it might be multidimensional, right? And so I really encourage you guys, if you haven't read this paper yet, it's great. I think this represents the de rigueur, thinking on how we should think, on the way evolutionary biologists think about divergence in allopatry. But the authors were using a one-dimensional Brownian motion as a null hypothesis. And I'd like to interrogate that a little bit in this talk. Another paper that came out somewhat recently that I really got worried about when I saw the title is this paper by Bolnick and colleagues on a multivariate view of the speciation continuum. Also a really cool paper, not exactly what I'm going to be talking about, but I do want to draw attention to the axis. Oh, it's getting cut off. That's a bummer. So their axes, they're including phenotypic divergence here as an axis that's just one-dimensional. But I think we all know that this is a multi-dimensional axis, but that might have implications for the way we think about how speciation could work. And actually, it was a huge bummer when I started working on some of these simulations and first started thinking about what was happening with random walks in multi-dimensions. Mike Landis actually brought my attention to this paper where Carolyn Tucker et al. Just found this pattern. They didn't talk about Polya's theorem of recurrence or transience, but this is essentially what they're talking about. Their motivation for writing this paper was to think about the link between phylogenetic diversity and trait diversity. So here they're looking at the relationship between functional diversity and phylogenetic diversity as you look at more traits. But basically these results are essentially the same as what I'm going to be talking about with my little simulations, but they just didn't apply it to the question of diversification, maintenance of species diversity, buildup of species diversity, divergence in allopatry, et cetera. So this paper essentially makes everything I'm about to say redundant, but I think it's the application of this mathematical property to diversification, I think maybe is somewhat novel. So my original goal is the reason I started trying to simulate divergence in allopatry was I wanted to know what the null hypothesis should be for trait divergence in a non-adaptive radiation. A couple years ago, my advisor, Rebecca Rundell, and I wrote a paper on non-adaptive radiations, and I thought it would be, usually the way people think about non-adaptive radiations is if there's a bunch of species that look exactly the same and ecologically are exactly the same, more or less, that's probably a non-adaptive radiation, right? But there should be maybe some wiggle room there. Like how different can they be and still be part of this sort of non-adaptive process. And just as a side note, I wanted to try to illustrate this in a more kind of whimsical, organism-centric way. So I sent this very terrible drawing to my friend, Brittany Ray Benson, and she gave me this nice little salticid drawing. She's obviously a much better artist than I am. But I wanted to try to represent the idea that organisms have, you can think of them as having like a two-dimensional geographic range space, and then a multi-dimensional population cohesion space in green, and then a resource use survival space in blue. And all of these things potentially have to be different for speciation to be maintained. Or at least if the population ranges overlap, then the traits have to be different. And all these traits could be on their own unique little random walks. And so this figure, I apologize for the axes, but it's on the x-axis, it's dimensionality in just ascending powers of 2, so 1, 2, 4, 8, 16. And then Euclidean distance on the y-axis. And basically, these are just random walks. There's no diversification here. This is just a pure mathematical process. But this is what happens after 10 time steps in green and 100 time steps in dark green. And these are simple random walks on the lattice. So all that means is that each time step you can take exactly one step of exactly length 1 in one sort of cardinal direction. So in a two-dimensional random walk on the lattice, you can only go up, down, left, or right each turn. Another tricky thing to disentangle is how we should conceptualize trait evolution. Is it, if there is a multidimensional trait space, is it a random walk that's a simple random walk? Or is it actually just like a bunch of one-dimensional random walks? Because that actually changes the behavior as well. So instead of the simple random walks, these are essentially 1, 2, 4, 8, 16, or 32 one-dimensional random walks happening at the same time. So what that means in 2 dimensions is just every time step you can go up and left, or down and right. You can't go up 2 steps, but you can, it's essentially the sum of these 2 random walks happening at the same time. And so just to contrast, after 10 time steps, the simple random walks in green, the n one-dimensional random walks in blue, just changing the y-axis here so that it's in log space to accentuate the zeros. But you can see that there's a lot more divergence in the n one-dimensional random walks. And then this is after 100 time steps. The green is the green is the simple, and the blue is the n one-dimensional random walks. So this is an actual simulation that I did where I was looking at diversification in a spatially explicit way, kind of a macro evolutionary simulation. Happy to talk about the details if you want to catch me later. But this is showing essentially the same pattern where it's root phylogenetic distance or time steps since divergence on the x-axis, and then log base 10 Euclidean distance on the y-axis. And here the colors have to do with how many traits I'm measuring at the same time. So all the light yellow colors are lots of traits at the same time. The dark purples are very few traits at the same time. And then this is just at phylogenetic distance 10, same data set, but just only at phylogenetic distance 10. Trait dimensionality on the x-axis and distance, trait space distance on the y-axis. And here you can definitely see a really distinct pattern of increasing Euclidean distance with increasing trait dimensionality. And just to switch back into a little bit of empirical data, there's a recent paper by Freeman et al. On the predictability of phenotypic divergence in geographic isolation. They were looking at species pairs of birds. And they saw that it seems like there is a relationship between divergence of these traits that are involved in maybe more in population cohesion than they are in sort of ecological divergence, and then a less predictable pattern in terms of the more ecological traits. Those of you who work on birds might argue that birdsong and plumage color might be ecological, but I thought this was a really cool paper that showed results that were really consistent with what I was thinking about. So this is, I wanted to look at species richness and how it varies with how many traits are important for diversification. And the main take home here is that at high dimensionalities, you can get a lot of species. This is, the colors represent species richness in each simulation. Each little box has about 600 different simulations that I ran. And then at low dimensionalities, if there's only if there's only the trait space for population cohesion and resource acquisition, if those are just 2-dimensional, then you don't get a lot of species based on the same simulation parameters. Same data, So the prediction could be that, all else being equal, trait dimensionality could be important for the buildup and maintenance of species diversity. It's really easy to break this pattern, though, just real quick. If you change the sort of minimum distance for allopatric speciation, the minimum sort of geographic range of the species, because species with tiny ranges, in my simulation anyway, diversify super fast, so you can override it really easily. Very quick aside about sphere packing, because MacArthur kind of, in his geographical ecology book, talked about this little cool analogy of species as crystal packing. I think that's really interesting. And then the poem that kind of inspired this, there's another pithy tweet about how high dimensional space can get really lonely. I think something that I really want to draw attention to is that we don't really know how this works in ordination space. And I think that's really important, because I think that's the way a lot of us think about multidimensional trait spaces, is by ordinating them. Ordination should swamp out Brownian motion in a lot of dimensions, but yeah, okay, anyway. Happy to talk about this more with other people, lots of people to thank. Yeah, maybe I can take a quick question or something. Yeah, thanks so much.